intestine is going to be our most distal end portion of our gastrointestinal tract of our alimentary canal. It's about 5 feet long and is about 3 inches wide, and we'll divide it into different segments based on its location within our abdominal cavity. So the main function of our large intestine is going to be finish any absorption that has not yet occurred as material has flowed through the small intestine. So this includes a little bit of water, as well as a few solutes like sodium and chloride ions, as well as a few vitamins like B vitamins and vitamin K. Now, the large intestine has a unique appearance that distinguishes it from the small intestine. It has something known as tinea coli, and what these tinea coli are these thin, long bands of smooth muscle that, as they contract, it helps to force and move along digested material in this peristaltic manner. So things are moving toward the terminal end of the large intestine via the contraction of these tinea coli. You also have these outpouchings of the large intestine known as haustra, or haustrum being singular, haustra being plural. And what they are, these little outpouching, these pockets of large intestine that help propel food along, as well as the tinea coli. And the reason that they form these pouches is due to the tension, this slight contractile state of the tinea coli. So I'll show you a picture of that in just a second. And then lastly, you have these little fatty appendages, these fatty pouches that extend off of the large intestine, known as epiploic appendages. And their function is not entirely known. They may be thought to just serve as an additional location for some adipose tissue to be stored. So their function is not entirely known, but we'll be able to see them. We'll be able to see them as a little small appendage hanging off the large intestine. Now, the very beginning of this large intestine, and really the beginning of our ascending colon, as we'll see, is the cecum. This is where the terminal end of the small intestine, so the ileum, joins with the large intestine. And then the appendix. We've all likely heard of the appendix, maybe related to appendicitis, is this small little worm-like structure that extends off of the cecum. So we'll take a look at those. And as we go throughout this last part here, this last lecture, we'll look at these 4 parts. So we'll look at the ascending colon, transverse colon, descending, and then our sigmoid colon. And as we discuss them, whenever you realize where they're at within your abdominal cavity, feel free to feel on yourself of where these would be. Now, you won't necessarily be able to feel them individually, but you can at least feel and follow along where they would be located within you. So, our large intestine is divided into these various parts here. So at the very beginning, we have our cecum, which is this little pouch-like segment here, as well as this appendix that's extending off. That then leads into our ascending colon. We have a transverse running transversely, and then we have a descending here along our left side. We then have this small segment known as the sigmoid colon, and that is followed up by this terminal end here known as the rectum. So here we can see some of those features that are very unique to the large intestine. So this small band, this narrow band of smooth muscle that you can see extending all the way up, as well as down here to the left, this is the tinea coli, and its contractile state produces these little pouches that we see all along, known as haustra or haustrum being singular. You can also see in various locations small little fatty segments that are extending off. So you can see a few here. You can see some up here in the superior aspect of this image. These are all of our epiploic appendages. So the terminal end of our large intestine ends as our rectum and our anal canal. The rectum is really just the last 8 inches or so of our large intestine. It contains these rectal valves that help to hold and keep the feces contained within the rectal canal. And then we extend a little bit further into our anal canal. This is leading to the anus, and we have 2 circular bands of muscle, 2 sphincters here that are under different control. So we're able to either control one and not control the other. So our internal anal sphincter is the involuntary muscle. So we are not able to control this. So it is able to open and close without our input. Now, the external anal sphincter, which I'll show you a picture of where this is located, this is under voluntary control. So we are able to control this, and thus we are able to control the times in which we defecate. So the mucosa in our anus will form these longitudinal regions known as anal columns, and we'll take a look at those. We'll take a look at those right here. So what we see here is we see these. We see the terminal end of the large intestine as the rectum. You can see these little outpouchings here that are kind of diverging into the lumen of this rectum. These are rectal valves. You can then see these little regions in here known as the anal columns. This is the anus at the very end. And then here you can see your internal and external anal sphincter. So internal is located more internally, and this is under autonomic control, so it's involuntary. We cannot control it. And then the external anal sphincter, we can control. We are able to voluntarily contract and relax this muscle. So just like the rest of our alimentary canal and rest of our digestive tract, we can look at it histologically and microscopically, and we can see various characteristics. So it will contain non-ciliated, simple columnar epithelium. So we see this in a large portion of our alimentary canal. So those cells will be a single layer of column-shaped cells, and they will not contain cilia. Now, with our respiratory tract, we have something similar. We have pseudostratified columnar epithelium, so still single layer that are generally columnar in shape, but those will actually contain cilia. But that's in the respiratory system. They also, in this region, we also have glands and absorptive cells that help to absorb what little material we need to still absorb at this point in our alimentary canal. And we still have goblet cells as well that will be secreting mucus. Now, our anal canal, as we saw in this picture here, so this region right in here, is going to contain stratified squamous epithelium that is non-keratinized. So if you remember, we also have non-keratinized stratified squamous epithelium in our esophagus, the epithelium lining our esophagus as well. We have keratinized stratified squamous epithelium that makes up our skin. Now, we also have bacteria in our digestive tract that is completely normal and is healthy and is fine for us to have it present. So what we call this is our bacterial flora, or our normal gut flora. And what it does is it colonizes our gastrointestinal tract, our alimentary canal, different regions of it, and essentially competes for nutrients, as well as helps to break down some indigestible material that we happen to ingest. So in that sense, it helps with digestion, as well as it has a somewhat of an immune effect, an immune function that you see with the immune system. It competes for bacteria and thus prevents the overgrowth of harmful or foreign bacteria. Now, we have to have bacterial flora, specifically some of them, in order to make different vitamins, and we'll focus on vitamin K. So vitamin K is necessary for the synthesis of various clotting factors that are produced in the liver, and it's made by this bacterial flora that we have. Now, if you think about this for a second, who might not have intestinal bacteria? Who may originally not, or at some point in their life, may not have this bacteria? So you may be thinking of newborn babies. So when babies are newborn, just as they're born, they have not been exposed to the environment yet, to their surroundings, so they have not had the chance to have their gut colonized by this bacteria. So if they don't have the bacteria that make vitamin K, you can assume that they don't have vitamin K. So what we'll give them is we'll give them an injection of vitamin K to prevent any type of bleeding event or hemorrhage. And then as they begin to live their life outside of the mother, outside of the uterus, they will eventually have their gastrointestinal tract colonized by by these different types of bacteria. So, as we are moving things within the large intestine, what we have is, as we said, we have a little bit of digestion and absorption occur here, but not really all that much. So as things are moving, we have these events known as haustral contractions. And what they are is those haustra that we looked at, they will contract and the tinea coli surrounding them will shorten and contract. And this produces mass movements, which are large movements of fecal material along the large intestine. And several times a day, these will move and be pushed into the rectum. And at some point, we will get something known as a defecation reflex, where these mass movements will push their way into the rectum, open the internal anal sphincter. If you recall, that is the involuntary one, so we do not have control over that. And then if we wish to defecate voluntarily, we will voluntarily relax the external anal sphincter, and we will get our bowel movement. So, in the in different parts of our gastrointestinal tract, we have this digestion occurring. And so the idea here is that things are being broken down from something larger into something smaller. So we're going from a polymer of some sort into a monomer. So if you think of our different macromolecules, you have proteins, you have nucleic acids, lipids, and carbohydrates. These will all be broken down by a specific type of enzyme. So for carbohydrates, these will be broken down by things known as amylases. So we have pancreatic amylase and salivary amylase. Our proteins will be broken down by something known as proteases. Now we have different proteases in our stomach. And if you recall, we have parietal cells. They will ultimately secrete stomach acid, so hydrochloric acid. That is necessary to convert pepsinogen into pepsin, and then pepsin is a protease. You also have a couple of these known as trypsin and chymotrypsin that break down proteins in the small intestine, and these will actually exist whenever they are moving throughout the pancreas. They will exist as proenzymes, and what proenzymes mean, that means they are the inactive form, so trypsinogen and chymotrypsinogen, and they will ultimately be converted into their active form to function as a protease. And then you have lots of these other ones, so carboxypeptidase, aminopeptidase, and dipeptidase. These are what we call brush border enzymes, and the brush border is the very apical region of our small intestine, the epithelium in our small intestine that contains the microvilli, and they reside within those microvilli and right along it. And as things pass over it, as these different proteins pass over it, they will work on the proteins to break them down into smaller free amino acids. So we also have, we had proteins and we had carbohydrates. We also have lipids being broken down by lipases. We have pancreatic lipase will break down our triglycerides into fatty acids, and we also have lipase in the mouth via the tongue, so we have lingual lipase as well. And then lastly, we have the breakdown of our nucleic acids, so deoxyribonucleic acid, which is DNA, and then ribonucleic acid, which is RNA. These will be broken down by pancreatic nucleases, and these will be broken down into their constituent parts, which is a nucleotide. That's the building block of nucleic acids, and a nucleotide is composed of a nitrogenous base, a sugar, and then a phosphate group. So here's a large list of different enzymes and what they act on. So we have a source of something, where it's coming from, the type of enzyme, and then what it works on. So you can see amylases are going to work on our starches or our carbohydrates. We have pepsin that we talked about working on proteins. We have different other ones working on proteins, our nucleases working on our nucleic acids, and then lots of these working on different sugars. So we have a few other hormones and other products that will be involved with the digestion and breakdown of different ingested materials. So gastrin is probably the most common and the most referred to hormone. So what it is It is produced by enteroendocrine cells known as G cells in the stomach, and sometimes gastrin is referred to as the get out hormone. And so, what this refers to is the various actions of gastrin promote material and substances leaving the stomach. So that's why we call it the get out hormone. It's promoting substances leaving the stomach. So what you have is gastric acid secretion. So you begin to break down more material in the stomach so that it can then be moved on to the duodenum. You have smooth muscle contraction, so to increase gastric motility and mechanical digestion in the stomach. You have the relaxation of the pyloric sphincter, which is the circular group of muscle leading from the pyloric region of the stomach to the duodenum. And then you have the relaxation of the ileocecal valve, which is down at the junction between the ileum and the cecum. When this relaxes, it causes movement of material from the ileum and the small intestine into the large intestine, and this thus makes more room in the duodenum for material to enter as it leaves the stomach. Now, we also have secretin, which will be produced by other enteroendocrine cells, but these are going to be in the duodenum, so the small intestine. And a lot of these these will be involved with stimulating bicarbonate ion release from the pancreas, stimulates bioproduction from the liver, and then inhibits gastric acid secretion and motility. So all of these have to do with things that are now occurring once food or once digested material has left the stomach and has now entered into the small intestine. We have a couple more. We have something known as cholecystokinin, which is sometimes abbreviated CKK. This is also produced by the cells lining the duodenum. It also involves or is stimulated to be released whenever material enters the duodenum. So release of bile from the gallbladder, release of pancreatic juices from the pancreas with those enzymes, and then the relaxation of the hepatopancreatic sphincter, which we also know we refer to as the sphincter of Oddi. That is the joining of the common bile duct and the pancreatic duct leading into the duodenum. You also have one last hormone known as gastric inhibitory peptides, sometimes just referred to as GIP. These are produced by cells in the duodenum, the small intestine, and its main function is to inhibit gastric acid secretion and motility. So this slows the rate at which things are being digested in the stomach, as well as slowing the emptying of the stomach into the small intestine. So this is a link. You're free to copy it and look at this online, but it includes lots of these various hormones that we just discussed on a separate page, and it goes into detail on exactly what they are, where they come from, and what their effect is whenever they are released. So that will wrap up our digestive system lecture. As always, if you have any questions, feel free